Hello and welcome everybody to my channel ominibioentrance, and today we will be seeing CSIR PYQs June 2023 Shift 2. I will be dealing with applied biology and methodology, DNA replication, protein synthesis and genetics. These are solved questions of part C. If you are new to my channel, please do like, share and subscribe. I have also uploaded old CSIR PYQs. You can refer to my channel for the same. Then next one. A researcher isolated a mutant of an endoplasmic resident protein folding enzyme PFE that has lost its KDEL sequence, that is endoplasmic retention sequence. Potential consequences of such mutation are given below. Which one of the following options represent the combination of all correct statements? So KDEL is the target peptide sequence located on the C-terminal end of the amino acid of a protein. Now KDEL sequence prevents the protein from being secreted from the endoplasmic reticulum, and also it facilitates its return back to the endoplasmic reticulum if it has been accidentally exported also. Now this is mutated in as per the is lost. KDEL sequence is lost as per the question. Now what will be the consequence? Now PFE is found in the extracellular space. So what will KDEL do? KDEL, it will inhibit the return of this protein to the endoplasmic reticulum and it will be secreted outside of the cell. So A is correct. So PFE is found in the extracellular space is correct. Then next one, PFE is degraded in the endoplasmic reticulum. PFE will be degraded in the no, it will not enter the endoplasmic reticulum itself. Then how will it be degraded? Unfolded proteins increase in the endoplasmic reticulum. That is also correct. PFE is transported to the cytosol. No, it is to the extracellular space. So A and C are correct. Answer will be option 3. Then next one, column X lists protein that play a role in mediating DNA recombination processes, and column Y lists the possible function of these proteins. Which of the following options represent all correct matches between column X and column Y? So RAD51. RAD51, it is for strand invasion. Then comes SPO11, that is for causes double strand breaks in meiosis. RAD52 and RAD59, it causes assembly of the strand exchange proteins. And MRXN complex, it is for resection of ends of DNA strands at double stranded break sites to create single strand overhangs. So your answer is option 3, A4, B2, C1, D2. Then next one, given below are approximate length of DNA fragments obtained on agarose gel electrophoresis following restriction digestion of a 3 kb circular plasmid with a different restriction enzyme. So these are the different restriction enzymes. We will see what we get after restricting the 3 kb plasmid with these different enzymes. Then based on the information, we have to see for the incorrect statement. So first one, BAM-H1 digestion. HINC2 ECO R1, they give 3 kb plasmid as such. So they are having only single restriction enzyme site in the plasmid. HINC2 and ECO R1. The BAM-H1 is having 2 sites. That is why you get 0.5 kb and 2.5 kb. So this is the BAM-H1 site. This is between two BAM-H1 sites. It is 0.5 kb and the rest will be 2.5 kb. 3 minus 0.5 is 2.5. Then next one was ECO R1 plus BAM-H1. Together they have digested. Now ECO R1 is having only a single site, that is 3 kb. We will put it outside the BAM-H1 sites. So ECO R1 BAM-H1, you get 0.5 kb. Yes, BAM-H1 will be released. 0.5 kb will be released. 1 kb and 1.5 kb. 1 kb here and 1.5 kb. This is upon digestion. You get these two fragments also. So one will be 1 kb and the other will be 1.5 kb. So 2 will be like this. Then eco R1 plus Hinc 2 you get 1.3 kb and 1.7 kb. From this we cannot make out where the Hinc 2 is located. Now if you see BAM, but this is the map of the digestion. I have just hypothesized that Hinc 2 will be located between two BAM H1 sites. Now BAM H1 and Hinc 2, if you see, you are getting 0.2 kb and 0.3 kb. That is, we can hypothesize again that Hinc 2 site is between two BAM H1 sites. So if you put Hinc 2 between two BAM H1 sites, you get 0.3 and 0.2 and a 2.5 kb hole. So Hinc 2 must be between two BAM H1 sites. Now you can confirm it by this digestion. Eco R1 and Hinc 2 you get 1.3 kb. Hinc 2 is here. So you get a 1.3 kb and a 1.7 kb upon digestion. If you do it, you will come to know. So these are the various maps upon restriction digestion. Then we will read the statements. Now we will see the statements. Based on the above information, which of the following statement is incorrect? Hinc 2 Eco R1 have single recognition site in the plasmid. Yes. They give only 3 kb upon digestion. So they are having only a single recognition site. Hinc 2 site is located between two BAM H1 sites. So Hinc 2 site is located between two BAM H1 sites. That is also correct. The distance between Eco R1 and BAM H1 site is less than that between Hinc 2 site and BAM H1. So the distance between Hinc 2 and BAM H1 is actually lesser than between Eco R1 and BAM H1 site. It is more. Eco R1 BAM H1 site is more. So what they have told is exact opposite of that particular statement is correct. So statement 3 is not correct. Hinc 2 is located closer to one BAM H1 site than the other. That is also correct. It is not exactly in the middle. So it is located to one BAM H1 site more closer than the other one. So 4 is also correct. So the answer here is option 3. They have asked for the incorrect statement. Two protein kinases K1 and K2 regulate an intracellular pathway in response to the extracellular signal. The following observations were made regarding the pathway. So suppose it is like signal, then K1 and K2, then you get a response. Now if we take it in that way, K1, K2 response, signal K1, K2 response. Now first observation. Response is observed even in the absence of an extracellular signal when a mutation permanently activates K1. Okay. K1 then it activates K2, then response. Now if mutation permanently activates K1, then K1 will activate K2 and you get a response. So that pathway might be correct. Now 2. Response is observed even in the absence of an extracellular signal when K1 contains an activating mutation and K2 has an inactivating mutation. Now, if it was K1, K2, then response, then as per 2, that is as per this one, response is observed even in the absence of an extracellular signal when K1 contains an activating mutation and K2 has an inactivating mutation. You shouldn't, now if it was K1, K2, then response, you shouldn't get a response because K1 is activating mutation and K2 is inactivating mutation. So if K2 comes after K1, then the response will not be observed because K2 is inactivating mutation. So even if K1 is activating, K2 is inactivating, you won't get a response. But here response is observed. So K2 must come in the beginning, then only K1, then response. So it has to be K2, after that only K1. So that is what you will see in case of 2. Now 3. No response is observed even in the presence of an extracellular signal when K2 is inactivated by mutation. Yes. Now if K2 is inactivated by mutation, you won't get a response because it is K2 then K1 then signal or response, then response, not signal, then response. So again it shows that K2 activates K1. Response is observed even in the absence of an extracellular signal when both kinases are activated by mutations. Okay. So which one of the following statement is correct? K1 inhibits K2, K2 inhibits K1, K1 activates K2 activates K1. So the answer here is option D. K2 activates K1.